Welcome to this session at Medical Sciences Made Easy by Naphtali Muhumuza. And in this session in hematology, we want to discuss different types of white blood cells. We want to see the morphologies, and we have seen it in an exam where students are asked to describe different morphologies of leucocytes, or what we can call white blood cells, as seen under Romanowski stained thin film. So we want to discuss different types of white cells under this session. And please always remember to subscribe, share your comments, and like the video so that we can continue to work together. So the first cell we want to look at this time is the neutrophils. And neutrophils, they are 10 to 15 micrometer in diameter, 10 to 15 micrometer in diameter, and their cytoplasmic granules, they are always fine. They have fine cytoplasmic granules, and these granules stain pink. As we know that neutrophil, the word neutrophil comes that it takes both the eosine and the methylene blue. So it is neutral, and it ends up staining pink in color. So it has fine cytoplasmic granules that stain pink under Romanowski stained thin film. And the shape of the nucleus is, it is lobed with 2 to 5 lobes. So the normal one has 2 to 5 lobes. And when I'm to draw it, find this being our neutrophil. It has 2 to 5 lobes, whereby these lobes are eccentric and they have clamped chromatin pattern. And the lobes are connected by a small nuclear thread. So this is the neutrophil, and we are saying it takes up both the eosine, the methylene blue, and the cytoplasmic granules are fine, and they are staining pinkish, which is a combination of eosine and the methylene blue. And these neutrophils, their percentage composition in blood is that in adults they measure between 50 to 75%. That is in adults. But in children, it becomes slightly lower. And their major function is that these guys are phagocytic cells. They play a very big role in phagocytosis. That is engulfing the bacteria that enter in our body. So they are responsible for engulfing. That is what we call phagocytosis. They play a big role in phagocytosis of any pathogens. And another major importance is they participate in acute inflammation. So when you go to acute inflammation, you find neutrophils are playing a big role in acute inflammation. So those are the major two functions of neutrophils: participating in phagocytosis, that is engulfing bacteria that enter in the body, and they are also responsible to participate in acute inflammation, which plays a big role in defending our body. So this is what we can discuss about neutrophils. Neutrophils meaning they are neutral because they are taking up both the eosine and the methylene group. We have seen it. Then the next cell we can discuss, the next one we can discuss is the eosinophil. This eosinophil, its size is slightly larger than the neutrophil, so it can be 10 to 16 micrometer in diameter. And the cytoplasmic granules, they are coarse. It has coarse orange staining, orange, red, granules. So these granules, they are coarse and they stain orange-red with Romanowski stains. So these are how we see them. And the shape of the nucleus is bispectacle in shape. You can call it bispectacle. It has two lobes. So it is bilobed, but with bispectacle in shape. So it is bispectacle in shape. And if I can draw it, it is slightly larger than the neutrophil. And it has a bispectacle shaped nucleus, whereby the nucleus is joined together by, and the nucleus always stained purple because it contains the nuclear material which takes up the methylene group. So we are seeing it stains purple with a cramped nuclear pattern, but being joined together by a thin chromatin. And we are seeing the granules, they are coarse. It has coarse granules which stain orange red. They are coarse. This one has fine, but this one has coarse granules. It has coarse granules. And that is the neutrophil. And these coarse granules of the eosinophil, its percentage composition in peripheral blood is 2 to 6% in blood or in peripheral blood. And this cell is also responsible for phagocytosis, whereby it kills helminthitic parasites like the hookworms. It can kill them. So it is also a phagocytic cell, but majorly targeting the helminthitic parasites, which is different from the neutrophil, which is responsible for the bacteria. Then another function of it that this also participates in allergic reactions. Allergic reactions also participates there. So these are the major two functions of the neutrophil as seen under Romanowski stained thin film. So looking at this, the next cell we can look at, because we know that leukocytes are divided into two, the granulocytes and the agranulocytes. So we have begun with the granulocytes, which are the bands. So we have the neutrophils, the eosinophils, then the last one is the basophil. The basophils, they measure between 10 to 12 micrometer in diameter, meaning among the granulocytes, basophil is the smallest. And its granules, it has coarse staining deep blue or deep purple granules. These granules are very coarse and they stain deep purple, and the shape of the nucleus is S-shaped, has S-shaped nucleus, or sometimes you can say indented nucleus. And this nucleus is always obscured, or it is always covered by the granules, because granules are very coarse and they obscure or they cover the nucleus, so it is hardly seen. And if I can draw it being the smallest in all, it has S-shaped nucleus or indented nucleus, but this nucleus is always covered by deep, coarse, deep blue staining granules. So it is hardly seen, because we see the granules concentrated at the nucleus, so it is not easily seen. So this is our basophil. And these granules, they release many inflammatory mediators we shall see the percentage composition in blood is less than 1%. And we are saying that these granules produce heparin, which is an anticoagulant. They produce serotonin, and they produce histamine. And these guys participate in allergic reactions whereby heparin for it is responsible for, it is an anticoagulant, so it stops blood from clotting. That's why our blood cannot clot, because of presence of the basophils, whereby these basophils in tissues, we call them mast cells. So in tissues, they are called mast cells, and they are producing heparin, which is an anticoagulant. And another function of serotonin, for serotonin, which is produced by these deep staining granules, is responsible, it is a strong vasoconstrictor. So for it enables the vasoconstriction of blood vessels. And then histamine is the one that triggers hypersensitivity. Hypersensitivity, or what you can call allergic reactions. So histamines. So the major function of these basophils is that we see they produce heparin, which is a natural anticoagulant in our body. They produce serotonin, which is a strong vasoconstrictor, so it causes vasoconstriction, narrowing of the lumen of the blood vessels. And we see also production of histamine, which participates in allergic, or what we are calling hypersensitivity reactions. So these are the major functions of the basophils can talk about. Then this is now the granulocytes. So we are remaining with other two, which are the agranulocytes. These ones that lack granules. Why do we call them agranulocytes? Because we do not see their granules under Romanowski stained thin film. And the first one under the agranulocytes, we have the monocyte. And these monocytes in tissues, we call them macrophages. In tissues, we call them macrophages. We say the basophils in tissue, they are called the mast cells in tissue. And these monocytes are the largest. They are the largest, which measure 15 to 22 micrometer in diameter. And about the cytoplasmic granules is that they have no granules when stained with Romanowski stains, and the shape of the nucleus, it has kidney, or it has bean shaped, or it has horseshoe shaped nucleus. And this nucleus also stains basophilic or blue. Then it lacks the granules, but the cytoplasm of the no granules, but the cytoplasm stains blue. And then if I am to draw the structure, it is the largest of all. And sometimes the outline is irregular, it has a regular outline. But the good news is that the nucleus is eccentric. It's a centric nucleus, but bean-shaped. It has the bean-shaped nucleus. Bean-shaped nucleus with clamped chromatin pattern and no granules in the cytoplasm. So this is the monocyte. And we have seen that the cytoplasm stains blue. So this is the structure of the monocyte, which is the largest. So even when you're drawing, you have to make sure that these drawings move with the sides. If we are saying it is the largest, then we should see it here. That indeed in the drawing we are seeing it is the largest. And in the percentage composition in the blood, we see it ranging between 2 to 10% in adults. And its major function, we see it is in phagocytosis. These macrophages in the tissue, they play a big role in phagocytosis by killing any microorganism It is responsible for iron metabolism. You see it participating in iron metabolism. And please, in science, I never say it helps. We do not use the word help in science. So whenever you are giving function, don't say it helps. It participates, it plays a role. And we see it also playing a big role in removing worn-out tissues, removal of worn-out tissues. And this monocyte, the same, it is responsible, or it acts as antigen-presenting cell. It acts as an antigen-presenting cell. How do I mean? What do I mean is that if this is the cell and the bacteria has invaded it, it engulfs it, and if I find the bacteria inside, it forms the phagosome to break it into small peptides, and these peptides are represented to the CD4 or to the T cytotoxic, to the CD4 cells by the major histocompatibility class type 1, depending if it is a virus, if it is intracellular, or type 2 if it is extracellular. And now, if this one is the monocyte, it is going to present a portion of the microorganism on the surface. So that's why we say it is antigen, it acts as an antigen presenting cell, which participates in humoral and cellular mediated immunity. So these are some of the major functions we can talk about. 4 of them: phagocytosis, iron metabolism, removal of worn out cells, and also acting as antigen presenting. So whenever you ask like 4 functions of monocytes, do not forget to list them. Then the last one we can discuss as we end this session is the lymphocytes. And these lymphocytes, they are categorized into what we call small and large, whereby small they measure 8 to 12 micrometer in diameter. Then the large, they can measure 14 to 18 micrometer in diameter, meaning this one is immature. Some of these small, you can call them B lymphocytes. These ones you can call them T lymphocytes. And the smaller ones, of course, they have no granules. They have no granules. But the cytoplasm stains sky blue. Cytoplasm stains sky blue, or you can say pale blue, or purple. You can call it blue or purple. And the nucleus, it has a round stalk oval nucleus. And this oval nucleus stains, it covers almost the whole cell. It covers 3/4 of the cell. So it has a scant cytoplasm, but this cytoplasm stains sky blue. And if we have to draw, you see it is small in size, and we are saying the nucleus has a centric nucleus which covers almost the whole cell, and it is round in shape. So this is the lymphocyte, which is small, and wholesomely all of them in percentage composition, they range between 20 to 40 in adults, but in children they range between 40 to 60%. That is in children. Then their major function, as I want to let me first talk about the large lymphocytes. For the large lymphocytes, for them, as we see, they measure between 14 to 18 or to 20. Also, they have no granules. They lack granules. Then they stain blue. The cytoplasm stains blue. Then the nucleus, it is still round. And for it, it does not cover much of the cell. It can cover 2/4 or half of the cell. So it does not cover much of the cell. And if we can draw it here, it is It is a bit larger than the small, and it has abundant cytoplasm. But sometimes it is eccentric with no granules. And their major function is that this one we are calling it B lymphocyte, this one we are calling it T lymphocyte. The B lymphocyte participate in what we call humoral immunity. How they are when they are in the tissue, they develop in the blood, they develop into plasma cells, and these plasma cells, they produce what we call antibodies. So they produce antibodies. Those are the B lymphocytes produce antibodies, or what we call immunoglobulins, which provide immunity, what we call humoral immunity. So they provide antibodies, or produce antibodies, proliferate into antibodies and memory cells. And these guys that even when you have suffered malaria, the body is able to keep memory, to know that when it comes back, it can easily fight it. That is production of antibodies and memory cells. Then also these lymphocytes, they also participate in fighting against viral infections. They fight or defend the body against viral infections. So whenever you have a viral infection, we can always see neutrophils when it is acute, but when it becomes chronic, they become depleted. So it fights against viral infections, and we see the T lymphocytes, they are responsible for cellular immunity. They participate in cellular, or what we call cell-mediated immunity, whereby these T cells, they are divided into three. The CD4 positive, the CD8 positive, and the T suppressor. Whereby the CD4, they help in opsonization. They do a big role in opsonization. That is tagging the infected cells. Then the CD8, they are responsible for killing infected cells. Then T suppressor is they regulate the action of the CD8. That when the CD8s are killing, they should not overkill to finish up even the normal cells. So this is what we can discuss about morphologies of different white blood cell, or what we call leukocytes, as seen in Romanowski stained thin film. And we have seen both the size, we have seen the cytoplasmic granules, and we have seen the shape of the nucleus, the diagram, percentage composition, and function. And whenever you ask in an exam, this is how we want it to be presented. You should discuss all cells under these headings. And remember, point to remember, that the monocyte is the largest, and in the tissue they are called macrophages. Don't forget that. And we have also seen that that the basophils, they are called mast cells in the tissue. Don't forget that. And it is the smallest among the granulocytes. So always remember that. And we have said the percentage composition changes. We have seen in neutrophils, it is adults, it is this, but in children, it changes. It becomes lower because children have higher lymphocytes and low monocytes. So it changes in children. And we have seen all the functions. Thank you so much for listening until the end. Always remember to subscribe. Always share your comments and like the video. Thank you so much.